On time. We will carry on with our world-renowned speakers. It is my great fortune to be able to introduce Olaf Sporns, our next plenary speaker. For those of you that don't know Olaf, he is the Robert Schaffer Chair, Distinguished Professor and Provost Professor in Psychological and Brain Sciences at Indiana University. Professor Sporns has made deep contributions to theoretical and computational neuroscience. Many of you may also know him as the author of the very well-known book, Networks of the Brain, and also Discovering the Human Connectome. Again, he has many, many awards to his name. I will not eat up his time by listing them all. Olaf, it's so great to see you, and we're very much looking forward to hearing your presentation. The floor is all yours.
Fantastic. Thank you so much, Josh, for the introduction, and good to see you from Mia, and great to be here. I really appreciate the invitation to talk and to present. I hope that you can see my screen and can also hear me. If not, please give me a shout.
We can.
Very good. So my perspective that I want to bring to the table here really is centered on what's been called earlier computational intelligence. The architecture of neural networks in real brains. And I might add that I have a history of working in physical intelligence also with robots, and maintained a robot lab for a number of years. And the way I used to introduce my talks as an old introductory slides was sort of that, you know, along the lines of what Carl mentioned earlier, that, you know, brain and body kind of cooperate in the service of information and in the service of creating of promoting survival of the organism and creating appropriate internal models, generative models, predictive models that are able to guide behavior in ways that are adaptive and productive for the organism. Under that umbrella, I will focus largely on the left side of this today as the instance of connectivity and what it contributes to computation and to intelligence. That topic has grown really tremendously over the last several years. So first, a little bit of an introduction into what is now called network neuroscience, the field that's forming at the intersection of network science and brain sciences, and then a few topics coming after that. In neuroscience these days, networks are very much mainstream. Connectivity used to be a very arcane topic that some of us pursued back in the 1990s, largely in a solitary fashion. But now connectivity and networks have come to the forefront on many different domains, on many different fronts. With model organisms ranging from the nematode C. elegans to the rodent brain, and then also the non-human primate, of course, and a lot of work done in human connectomics, human network science, and down here in multiple domains. This is a picture from one of Carl's earlier papers, and I will come back to that interaction of structural and functional connectivity later in my talk. I think this development towards networks is driven in part by technology, by the availability of ever more voluminous, large, big data sets that come at us from all different measurement domains, from interaction networks among molecules all the way to behavioral and social networks that unfold between organisms and individuals. The main part of my talk will be in this middle ground, the types of networks we construct from observations of brain anatomy and physiology and function as the brain is active and engaging in behavior. And that's generally now called connectomics. Last introductory slide, there's a confluence of different methodological tools, from molecular screening tools to anatomical mappings, high density functional recordings, and behavioral data onto a common construct, which is collections of nodes and interactions or edges, networks in other words. And this confluence of different data streams onto a common domain of mathematical modeling and statistical modeling has given rise to many new interactions between these different domains of biology and neuroscience that play out in the context of network neuroscience. Some of the network tools that I'll mention down below, the notion of distances in a graph play an important role, but also modularity. It's been mentioned a couple of times today as a way of formalizing the notion of functional specialization. I'll come back to it because it's a fundamental aspect of how real brains are actually organized, we think. A couple of words here about what we know about the human brain at this point. There's not enough time here to go into any amount of detail, so just a few tidbits in a very small format. Until quite recently, our knowledge about the human brain came largely from post-mortem dissection of specimens that were dissected and then photographed, as in this image here. We're literally seeing the wiring extending between remote regions of the brain through the interior part of the brain we call white matter. Modern technology now allows us to infer these trajectories of long-range pathways between different brain regions using non-invasive neuroimaging technology called diffusion imaging and tractography. The technology going into this is very complex, involves many steps, and is still being refined as we speak. So the kind of data we get at this point are really preliminary, I would say, and subject to revision as we move along and this technology becomes more and more refined and sensitive and accurate in the way it maps human brain anatomy. The output of such a network analysis is in fact a lookup table of what's connected to what, really a large matrix. Matrices are mathematical objects that we can analyze with the tools of graph theory and network science. And they record in a very handy summary diagram really basically what's connected to what, indicating presence of connection in this matrix by a non-zero element. And the strength of that connection is indicated as well. What we're seeing here is a very early map of the cerebral cortex of the human brain that we created a few years ago now with very few subjects at the time in our study, only 5. Many subsequent studies have been carried out in virtually every imaging center on the planet, looking at the architecture of human connectome networks in different cohorts and with different measurement techniques. The kinds of features that we encountered in our early work have largely been replicated across these different cohorts. I want to briefly mention what they are. It turns out every brain region has its own unique set of network relationships to the rest of the brain. We call it a connectivity fingerprint. It's a unique set of inputs and outputs. And if you are a member of a network, what you are capable of, the contributions you can make to the overall functioning of that network really are enshrined in the kinds of relations that you maintain with your neighbors now in a topological sense. So that notion of connectivity fingerprint looms large, I think, in our field still today. We find that in most, if not all, data sets we have looked at and others have looked at, that there are it's usually a small admixture of brain regions, neurons in some cases, that have high degree, have a large number of connections, larger than the average. And we would refer to that as a broad, perhaps heavy-tailed, perhaps log normal in some analyses, degree distribution. There's high clustering and short path length. In other words, we can move information around in these networks with a relatively short, perhaps surprisingly short number of links on average. And there is evidence of modularity, modules that are densely connected clusters of brain regions and neurons that are interlinked, mutually connected by a small number of important interlinking nodes that we call informally hubs. These hubs also tend to interconnect with each other to form a structural core, sometimes called a rich club. Those types of connectional features are what we encounter in human connectome data, and as we see in a moment, we also encounter in other animal brains that have been subjected to similar type of analysis. There's a huge amount of activity in the field these days. One area of emphasis really is, and this has to do with applications of connectomics, is in individual differences. How do individuals differ in terms of their architecture, their cortical anatomy, and how do those differences relate to differences that we might observe in behavioral capacities, cognitive operations, etc. We haven't, at least I have not heard much today about individual differences in the context of embodied intelligence. I wonder if that might be a topic for discussion. Do we care about them? Are they important? In medical applications, individual differences loom large because there is no such thing as the human brain. Every brain is different. And in fact, the architecture of brains is significantly different from one individual to another. And how that plays out in behavior and cognition is one of the major emphases of research in my field. A few words now about model organisms. The arrival of modern imaging techniques, reconstruction techniques, all the way from electron microscopy to non-invasive imaging in humans, has allowed us to assemble a number of connectome data sets now, ranging from invertebrate species such as the nematode C. elegans and Drosophila, to the avian brain, to other mammalian species, rodents, non-human primates, and of course humans. And that sets up an opportunity for us to compare these different architectures to each other. How are they? How do they differ, or asked differently, what are the universal features that underlie them? In this context, I want to just briefly mention the history, in some ways our deep history in our field here, going back all the way to the early days of cybernetics, and some of the pioneers of that era were already intrigued by the topic of connectivity. Gray Walter, in this little diagram on the left hand drawn, apparently, is speculating on what might happen when we take a single neuron and we start enlarging a nervous system to have 3 neurons, 5, and many more, 8 neurons. What happens to the connectivity between these neurons? How does that connectivity pattern proliferate? He talks eloquently about how connectedness might in fact be the key to understanding cerebral function. So there was a time when this was already bubbling to the surface, but not much data yet at the time, certainly. Much more data available now, much more opportunity for us to dig into those principles in greater detail. Over the last decade or so, a number of organisms, as I mentioned, have been mapped, including Drosophila, including non-human primate, and the rodent brain, of course, important model organism in neuroscience. And some of the common themes epitomized here in these illustrations are modular organization, the existence of somewhat dense clusters of brain regions or local processing units, as I called in Drosophila, that are densely connected internally and less densely connected between each other. The existence also of important nodes that connect these modules to each other, so that connectivity is not lost across modular organization. And these are sometimes called hub nodes. And on top of that, in the case of larger scale analysis of nervous systems, the existence of clubs or cores, which are associations of hub nodes with each other, dense connectivity patterns between them that go above and beyond what is expected by chance, and might indicate a potential infrastructure for sharing information and transferring information between different internally dense but otherwise sparsely connected communities. Importantly, these patterns repeat on multiple scales in terms of neural architecture, from brain regions all the way down to circuits. To me, this was a surprise, to be honest. I did not expect some of these features to be found even in single neuron networks. Shown here examples in the rat hippocampus and the macaque cortex, recorded with multiple invasive electrodes in behaving macaque monkeys. What we're seeing here is that, first of all, using transfer entropy as our way of mapping information flow or information transfer between individual neurons, that not all neurons have the same number of incoming and outgoing connections. Some are privileged in the sense that they have more than others, informally called hubs, and these are interconnected more densely than expected by chance. Interestingly, now we're engaged in ongoing studies with collaborators in Germany. We're finding evidence that these affiliations between neurons change on a fast time scale of hundreds of milliseconds, perhaps indicating different patterns of information flow that are deployed in the context of ongoing behavior. So the link to behavior and to action in the environment is something that intrigues us a great deal. But again, bottom line, we're finding similar types of connectivity, modular hubs and cliques and clubs in microscale single neuron anatomical arrangements as well. Just as a methodological aside, I mentioned multiple scales. This is a topic that looms really large in our field. When it comes to assessing the architecture of a nervous system, it's important to know what scale you're operating on. A lot of the studies involving whole brain analyses still refer to aerial arrangements, brain regions, entire parts of the brain that are, a centimeter across and contain millions of neurons and are averaged into a single representation of a node. But we also, of course, can map the brain at other scales, microscales of individual neurons. That is a daunting task in the case of humans with the 80 or so billion neurons that make up our cerebral cortex, for example. But we have methodological tools available to us through network science methodology to actually assess modularity on multiple scales. We're doing this using global maximization of a modularity cost function, extracting modules across different scales, and then smushing them together again into what we call a multi-scale co-assignment matrix. Essentially, for each node pair in the network, we are expressing the affinity that this node pair has across multiple scales of being affiliated in the same module. So functional specialization, which I think is a concept worth mentioning in the context of modularity, plays out on multiple scales. And I will come back to that in just a moment when we get to functional connectivity. Just a brief mention of a few details in the work that we're currently engaged in. Been a long relationship, collaboration with Larry Swanson on mapping different parts of the rat nervous system. What you see here on the upper left is essentially a rat brain that's been flattened. Imagine you're pancaking it on a lab bench. The outer parts here, the cortex, basal ganglia, thalamus and hypothalamus, midbrain and hindbrain, subdivisions shown in a symmetric arrangement. So that's the rat brain in 2 dimensions, if you wish. And we've been gradually making our way into mapping connectivity based on extant literature on rat connectivity coming from the histological domain, from the outer parts, from the cortex into basal ganglia, and then midbrain, and finally now endbrain and hindbrain subdivisions of the rat. And there are some interesting lessons that emerge. First of all, there's a fairly large number of brain regions we are already considering just in the forebrain alone, 466 on both sides of the rat. And when we look at these co-classification patterns, the fact, sort of capturing the propensity with which 2 brain regions might appear linked and affiliate with each other in the same network community, what we're seeing is, yes, we're seeing multi-scale patterns. We're seeing nested organization, shown here on the left, or rather on the right, in a classic sort of dendrogram representation. These are the 2 hemispheres of the rat brain. They are identical to each other. And what we're seeing now is a nested arrangement of subsystems that are modules, functionally specialized, that are starting to branch and then subdivide further, forming a hierarchy of functional specialization, of modularity based on connectivity alone. If we're zooming in on one part of this diagram, this is shown on the next slide, and drawing on the extensive knowledge that someone like Larry Swanson has about brain behavior and anatomy, we can affiliate many of these modules and anatomical communities with particular functional or behavioral connotations that are annotated here on the left. An example of how we can link a structural description of a circuit, such as the rat forebrain, in this case, the union of cortex, basal ganglia, thalamus, and hypothalamus, into functionally meaningful, behaviorally meaningful subdivisions that can be mapped onto known behaviors. That is reassuring to know, because these structural network-based decompositions of such a large graph into modules, they ought to have some functional relevance, and in fact they do. One note of caution, one lesson that I've taken away from studying the rat brain and its different subdivisions in great detail over the last few years is that different subdivisions do not all look alike with each other. A lot of emphasis in our field has been on cortex, in part because it's the outermost part of the brain, accessible with many of our invasive tools and with imaging, and most openly, flagrantly, one might say, affiliated with intelligence, and certainly in primates and humans. And the subcortex has received much less attention in some ways. But subcortical divisions, such as the hypothalamus, the thalamus, now we are in the midbrain with tectum and so forth, it turns out they have different topological features. So yes, modularity seems to be preserved largely, even going into structures such as the hypothalamus. But things like small worldness, for instance, are not encountered in some of these other subdivisions, and or club-like associations between hubs are not encountered. So there is actually some heterogeneity in the way that these architectures play out and are realized in different subdivisions of the central nervous system, probably related to different objectives that they support in terms of information processing, information storage, what have you, directing behaviors. But there's also probably a role to play, there's a role that is played by spatial embedding. And I want to emphasize this point for a few more minutes here. Why? Because we have to understand that the brain is a physical object. It shares that feature with the body, the rest of the body. It is physical. It is built from cells that have physical dimensions, lengths, energy demands, and that take up valuable resources such as volume. In the case of the nervous system, conduction delays play an important role because conduction is not instantaneous. It incurs a delay that's quite significant across distances that are typically spanned by brain circuits. So like in circuit design and like electronic circuit design, I'm superimposing that here in the back, there's an aspect of geometry and physical constraint that needs to be taken into account. And that is often forgotten, I think, by people who think of the brain as being somehow optimal with respect to some performance criterion, or perhaps with respect to intelligence itself. And yet, I suspect that it's not optimal in the strict sense, as many circuit arrangements that we could theoretically dream up in a simulation, let's say, are simply not physically realizable. As I sometimes like to say, if your brain does not fit into your skull, the chances of survival are somewhat compromised. So there's a physical aspect to the construction of a brain, geometry, but also energy demand, that really cannot be contradicted by the design process. The design is subject to those constraints. These are very important constraints that play a role. And that has led me with my colleague Ed Bullmore years ago to talk about the real topology that we find, which we call complex in the sense of being an admixture of both random features and regular features of topology. The topology that we find is sort of a trade-off that sits in the middle between two extremes. On the left, an extremely economical brain, a brain that has its wiring cost minimized. All wires are short. That makes it cheap to construct and to maintain, but it is not an optimal arrangement, as you can see right away, if you want to move information from one end to the other. On the other side, a brain that in a trivial sense can share information liberally and freely and with optimally short path length. However, it has lost all functional specialization. So that trade-off between a drive of the brain to be cheap and on the other side move information around with high efficiency and short path length leads us to that compromise in the middle, where many design features are directed towards keeping connection lengths short, such as modularity, while other design features, such as the existence of highly connected hubs and their interconnections over long distances, are directed towards moving information around more freely. So these two opposing trends in some way define where this trade-off ends up. I'd like to emphasize again that an astronomical number of architectures that we can think up in simulation or in thought experiments are simply not possible to physically implement and therefore play no role in biological evolution. So we have no way of telling whether the architecture we currently carry around with us is in any way optimal with respect to an abstract objective function. And so I like to think of this as a slight pun here on the topic of the conference as sort of a manifestation of embedded intelligence, embedded in the sense that it's geometrically and physically embedded in an actual circuit that has to be built and maintained. So that interplay of cost and efficiency, perhaps in a manifestation of the embedding of intelligence in an actual physical and biological circuit. So now let's turn over to what the brain is actually doing. So far, I've talked only about structural connectivity. Everything I've said so far really only applies mainly to anatomical circuits, wiring diagrams, as we sometimes call them informally, that refer to axons, synapses, neurons connecting with each other, the physical stuff that our brains are built of. But now now let's talk about functional connectivity, which a long time ago, a quarter century, I think, was defined by our own Carl Friston as a pattern of statistical dependence between often remote neuronal events that might unfold in different parts of the brain, different brain regions, or different populations of neurons. The statistical dependence is the key term here. It is a very different construct from structural connectivity, which refers to a physical entity. This little movie on the right here is, I think, quite thought-provoking. It's actually a projection of an MR activation pattern, bold activation pattern, MR signals recorded from a person in a scanner who is doing nothing in particular, just being awake and alert and going about their own mental business. We call that the so-called resting state. That's a very popular way of probing brain activity in our field these days because it requires relatively little instruction, can be carried out in virtually any population of individuals, and has turned out to be quite informative about how the brain is organized. I play this here because this is disembodied intelligence in some ways. This is a person in a scanner who's been prevented, who's prevented from moving, okay, who has been instructed not to move. And this is ongoing mentation. And we all know that we engage in this ongoing, mind-wandering mental activity quite a bit. And our brain is quite active when we do nothing at all in the environment, nothing overtly directed at a goal in our environment or any particular bodily movement. And we can construct from such time series, recordings of neural states, we can construct statistical dependency patterns, and those are functional connectivity. I want to emphasize here how important this resting state really is. It appears to be ubiquitous in the sense that it is found in every individual that we've ever looked at, and it consumes a large fraction of the brain's metabolic energy supply. In fact, more than half, perhaps 80%. So what is this ongoing spontaneous activity doing? Does it have a role to play in embodied intelligence? I'm wondering. I think, might be a point for discussion. A lot of the emphasis in embodied cognition, as far as I know, has been on sort of stimulus-driven sensory motor activity, engagement with the environment, doing something in the environment, while a lot of the energy that our brains consume, and it's a lot, is apparently taken up by ongoing spontaneous sort of background activity, which is something that is perhaps puzzling. Now, resting state networks have been studied in my field very widely. What we do is we take a movie like this, chop it up into frames, cross-correlate, very simple, just create a cross-correlation matrix, which is often called functional connectivity, and which gives us a very reproducible pattern. This is remarkable given how unconstrained the resting state really is. We can reproduce this pattern across individuals fairly well, across time and across different subject cohorts. And the blocks that you see in this matrix are coherent brain systems that are undergoing internally coherent fluctuations across time. They go up and down together in terms of their activation status across resting state. They have names such as default mode, dorsal attention saliency system, somatomotor system, that have connotations with respect to the kind of cognitive operations that they are engaged in and participate in humans. Again, very consistent patterns across multiple studies of large cohorts of individuals on different continents in different parts of the world. Is this notion of resting state networks, which really are modules in functional connectivity, nothing else, functionally specialized subsets of regions, has given us a totally new way of mapping out brain topography in humans and in other species as well. In our own computational work, we've been simulating functional connectivity for many years and right up to the present day. And here's an early movie of what we've simulated in the macaque monkey in terms of its ongoing, potentially ongoing simulated fMRI activity that might actually play out. And it was these simulations, these computational studies that gave rise to the idea that functional connectivity itself is not static or stationary, but instead varies across time. If we step a window across time in such a simulation, and we calculate various topological metrics of how nodes are interacting with each other, their pattern of connectivity, that pattern changes quite significantly across time, even in the absence of an external stimulation. There's an ongoing fluctuation of network states that seems to be ingrained in spontaneous activity as we observe it in individuals, in humans and other species as well, and in simulation as well. This work has been taken forward by people like Gustavo Deco and Victor Yerza, colleagues of mine that have done a lot more than I have on characterizing these patterns of dynamic functional connectivity and associated them with topics such as multi-stability, metastability, near critical behavior. In general, tracking fluctuations between more segregated and more integrated network states. That is the topic I want to finish my talk today, because it's something that I have maintained interest in over the years, and I feel it speaks quite directly to this notion of functional specialization and how it fluctuates and how it might support computational intelligence in the service of adaptive behavior. Very old work here that I'm still quite fond of, carried out almost a quarter century ago, on constructing multivariate information measures that can be deployed in a network setting to quantify and measure the distribution information across multiple nodes and elements. This is finding applications in embodied systems as well. Nihat Ai's talk earlier was a wonderful demonstration of that. I think it has many applications in that context. In our own work, what we did is we simply optimized through rewiring of connectivity patterns in an evolutionary setting, evolutionary algorithm, try to optimize various quantities such as multi-information and complexity. When we optimize complexity, we end up with a basically modular architecture, which in hindsight makes a lot of sense, because complexity is associated with reducing degrees of freedom down to a number that is sufficiently large so that activity can vary across time. So it shouldn't be 0 or 1. But a small number, while at the same time simplifying system dynamics significantly, sort of encapsulating, constraining degrees of freedom down to a few major modular components that can then fluctuate against each other and provide a repertoire of states that fluctuates between synchrony over smaller versus larger assemblies and integration and segregation, which is the readout of modular architecture. Another thought diagram here, the way to think about it is that functional connectivity and the way it changes across time fluctuates between more segregated states, which are, you might call them more modular, and more integrated or flat states where information is more freely shared between larger parts of the brain, perhaps called today low modularity states. In empirical work, we have investigated whether these fluctuations actually do occur in functional connectivity dynamics in the human brain. In fact, they do. Working with my former postdoc, Makoto Fukushima, he set out to investigate using a windowing approach how patterns of modularity change across time in humans in the resting state, constructing a null model to reject expected variations of modularity across time and retain only those that exceed the statistical null, and then looking at those episodes, lumping them together, creating average patterns, Some things stick out at us. The high modularity versus low modularity periods have recognizable topology and topography. So they're not arbitrarily modular in the sense of, just any modularity that we might encounter, but they have very particular signatures or fingerprints in the human brain. Very interesting to me is that when we have high modular episodes in a human resting state, what we're seeing is a dissociation of two sets of systems that we might broadly describe as those directed towards intrinsic processing and those directed at extrinsic processing. By intrinsic processing, I mean systems like default mode and control that are more engaged when we are mentalizing, retrieving memories, anticipating the future, and evaluating future reward or whatever, social cognition, while we have systems on the other side like visual, somatomotor, auditory systems that are more directly engaged in sensory processing and attention demanding applications of or attention demanding demands in the environment. So extrinsic systems and intrinsic systems dissociate or decouple from each other reliably, reproducibly across time as people engage in spontaneous mentation at and high modularity episodes. And then we have a more heterogeneous bag of states in between that are characterized by fluctuating, less modular arrangements, more integrated overall, that we can also track across time. In the last few minutes, just a few words about the line of work that I'm very excited about that we've started with our colleague Rick Betzel and my grad student Josh Feskowitz just recently. I mentioned functional connectivity as being a cross correlation. In fact, most functional connectivity studies are still carried out using essentially Pearson correlation of node time series as their summary metric of how statistical dependence is described and measured. What we've proposed is that we can unfold the Pearson correlation, unwrap it in time, essentially taking bold signals that are z-scored, normalized, and multiplying pairwise time series together to create what we call co-fluctuation time series, which are now specific to each node pair ij. And the average of this is exactly equal to functional connectivity. So the field as a whole has been focusing a lot, and believe me, thousands of papers have been written on functional connectivity to this point, has largely focused on the average, the global average of all these interactions across time, leaving us with just one number that we associate with a given connection between two brain regions. However, what we now do is we unwrap it, we take the one step back, leaving us with an exact decomposition of functional connectivity into its blow-by-blow account, into its momentary contributions. This is an exact mathematical equivalency. We can then map this out across the whole brain, and now we have I think, wonderful tool for actually looking at fluctuations in how brain systems interact with each other. The whole metastability, multi-stability framework is now accessible to us like never before. And I'll show you a demonstration of that in just a moment. And when we do that, one thing that we now end up with is an edge by time matrix, which is a representation of all the brain does across all its connections at any, over a long period of time. We're seeing these vertical streaks. These are burst-like events that have a high co-fluctuation, as we call it, high amplitude signals in the brain that are burst-like and intermittent. This is, again, a person lying in the scanner at rest, engaging in spontaneous mentation, introspection, what have you. That time course is punctuated by these high amplitude co-fluctuations that engulf much of the brain and have a reproducible signature, by the way, which is reminiscent of the dissociation between extrinsic and intrinsic systems I mentioned earlier. We have produced at this point some very, very dense data papers on this. I have no time to go into any detail. You're welcome to take a look at this. What excites me is we can actually now, in much greater detail than before, disentangle what happens sort of in a moment-by-moment account in terms of the brain-wide networks that unfold. This approach is can be adapted to information theoretic or other measures of statistical dependence that we might be interested in. It can, of course, be adapted also in an embodied context where the time series are not coming from brains or from neurons, but perhaps from actuators in the environment, from movement patterns, etc. So last, I want to show you a movie. Just to give you a first-hand account of what this kind of looks like. What you're seeing here is a resting state scan measured with fMRI, where we're seeing our edge by time matrix gradually accumulating. This is the time base here is about equal to seconds. This is about 15 minutes of time we're looking at. Up here, we're seeing the brain mapped out into a matrix of 200 by 200 brain regions, all interactions captured. Red and blue refer to positive and negative co-fluctuations. The little squares you see here are our brain systems that I mentioned earlier: visual, somatomotor, dorsal attention, ventral attention, the limbic system, frontoparietal, and default mode. And the upshot of this movie is that these systems don't really exist. Don't quote me on that. They don't really exist in a sense that they have objective, boundaries around them, and we can point to them at any moment in time, and there they are. They do manifest occasionally. So you see that blockiness appear in this matrix in very short, intermittent intervals. So the way we're thinking now about functional connectivity as a dynamic process is that these systems that have been mapped in human brains now for over a decade come into existence only during brief intermittent episodes and then dissolve. And they come into existence in different combinations, different modular arrangements, we might say, different patterns of functional specialization that are time variable and that have some repeating statistics to them. We have, of course, dug into this, as you can imagine, in great detail already. And they vary between individuals as well. But this new approach has allowed us to actually visualize this for the first time. Now, I come to the end, and I want to finish with a few thoughts about what the things I've talked about might contribute or not, or what opportunities that they might open in terms of research in the area of embodied intelligence. So my first thought is that, brain-body environment interactions are obviously central to the whole field. In my thinking, and thanks to my exposure to working in this area early in my career, I totally get that. I take that on board. There is a lot happening in this space. And I would put on the table that network modeling tools, perhaps using measures or metrics of information flow, information transfer, information decomposition, are very good at tracking patterns of these of tracking patterns of dynamic interactions across different elements in a body-brain system and environment. Comparative connectomics has provided us with some common but also some divergent themes. There are commonalities that I've mentioned, modularity among them, existence of highly connected brain regions or highly connected neurons. But there's also for sure divergent themes as our brains in different organisms, of course, interact with different body plans, different musculature, different behavioral patterns, different body sizes, what have you. And so there is perhaps some important links to be explored. How do architectures differ in the connection, in conjunction with the morphology of body plans and behaviors that result from that interaction? Multi-scale organization is important in our field: integration, segregation, dialectic, if you wish. High-low modularity states are gaining in importance in our way of assessing how individuals differ and how they process information. There's a common underlying theme here, which is, to me, complexity, which I still think of as the conjunction of segregation, integration across time and across space. And there's important differences that I see, perhaps overstating those differences, but deep nets or machine learning applications really pay no attention to complexity at all, and they perhaps it's because they are implemented in a geometrically permissive environment where there is no space constraint, no resource constraints. multi-layer machine learning networks can be as big as they want to be without any decrement apart from computational load. There's no real sense of geometry or cost or energy that needs to be consumed by the actual network itself. So there's architectures that are possible to achieve with machine learning implementations that are really not possible in the real world. I think complexity plays a role in that context in real nervous systems. The important role of resting state or spontaneous dynamics, something that came as a surprise in our field, really, which was largely dominated by people studying task and task engagement. Resting state was very prominent. I think the pendulum is swinging back a little bit, back towards task or more ecological settings within which people are observed and measured. So certainly the dialogue between intrinsic and task-evoked activity and dynamics is important in neuroscience. How important is that in embodied intelligence? Can we conceive of spontaneous dynamics as playing in spontaneous dynamics in the body as well as in the brain as playing an important role in constructing and designing adaptive embodied intelligence systems? And finally, the role of the body in generating information. This was something that I pounded on a lot when I was still doing this kind of work with robots. The notion that the body is one way by which the brain can feed itself information, can actually create information that would not be present otherwise in the environment. That interaction, of course, playing a prominent role in many of the participants of this conference's work, I think is fundamental. And one thing that we in neuroscience have not addressed is how does functional connectivity, how does it really get shaped by ecologically correct embedding of people in their environment. We're studying them in scanners for the most part in non-ecological settings. But what if we had a portable scanner and could actually look at people's brains as they are engaging in ongoing physical activity and moving their sensors around and their effectors in the environment? What would that look like? I think it's completely understudied and underappreciated in my field. So that was my my perspective. I hope it's useful, and I look forward to your questions. And thank you again for allowing me to be a part of this conference, and from seeing so many of my old friends and colleagues that I used to see a lot more of back in the day, you, Josh, and Fumia and others. So thank you for having me.
Thank you so much, Olaf. That was fantastic. And I will paraphrase a chat comment here. That's so cool, I can hardly stand it. I will agree. That was awesome. Thank you so much. As usual, there are a ton of questions here, so I will admittedly call on a few of them in a biased manner. You mentioned spatial scales several times. You showed some temporal changes over scales of minutes. What about development? Have you or others looked at putting, short time span animals in the scanner and look at how these networks change over developmental timescales?
Yes, I mean, we have not personally done that, but there's many people in the field are interested in development in humans, particularly big topic for many reasons, including applications in disorders and conditions and so forth. Big topic, yes. So people have looked and find systematic changes across development. One of the broad patterns that we see is that the brain tends to be somewhat more modular and more disconnected, as it were, at earlier stages in the lifespan. And then gradually connectivity becomes more dispersed, more distributed later on. That seems to be a global trend, both in anatomy and in functional connectivity.
Interesting, thank you. I see that Nihat has his hand up, and as a speaker for today, you may be able to unmute yourself. You want to give it a try, Nihat?
Oh, it works. So thank you very much, Olaf, for this great presentation. So in the first plenary talk today, your work on self-function, if I remember correctly, in that work, you had the TSE complexity maximized as a result of that self-structuring process, but it applied to the sensors. So that was a setting. The robot had a camera, and so this was the TSE complexity applied to the characteristics of the pixels of that camera image. And in your presentation, you showed us the basically high TSE complexity. You were referring to high TSE complexity within the brain. So is it a coincidence that at the periphery you have high TSE complexity as a result of self-structuring, information self-structuring, and internal complexity is also with the same measure high? Is it a coincidence, or do you think there's some connection?
First of all, I'm amazed that you can recall that work. I haven't forgotten about it myself almost completely. That was so many years ago. Yeah, absolutely. And I remember running these experiments with the robots in the lab and looking at this. No, I don't think there's a coincidence. I think that complexity, and this is a perhaps philosophical point, that the need for having complexity in a control architecture and an internal control architecture, computational intelligence architecture, the need for that, I think, has to do with the fact that our surrounding world also has structure on multiple scales, on multiple temporal and spatial scales, perhaps to which those control architectures have to adapt, perhaps in the service of what Carl called, creating a viable internal generative model. I think I'm very, attracted by that notion. I don't have the mathematical sophistication to follow this in detail, but I feel like that is in the same spirit. Somehow there's a matching between what our brains do and how our environments are structured, the physical reality we face. Also our bodies, the way our bodies move, the time constants involved in that, they impose limits on what brains can and cannot do in a given amount of time. So I think there's matching across these different domains and coevolution, obviously, and I think it's not a coincidence.
Mm-hmm. Thank you very much.
Thank you, Nihat. I think we'll take one more question for Olaf, and then we'll switch to the discussion session. Question in chat here from Andres. How does the connectome change in relation to body structure? Having limbs versus not having, maybe to animals that move with more or less degrees of freedom or sensory motor loop complexity?
Yeah, that's a great question. One of the exciting new projects we're just starting up is a collaboration with people who have actually measured connectomes in many different animal species across the mammalian family, at least, including many bats. Bats are mammals. There's many of them. They have very different, very intriguing biological behaviors and social interactions, etc. And so we have actually now, literally on my other computer here to the right, I've got lots of bats. I have a ton of rodents. I have a lot of primates, and we have dug into how do connectomes differ between these different species, and we are indeed intrigued by, can we relate those differences to differences in body plan, in sensory and motor capabilities. Body size plays a big role. I mentioned this earlier, even Gray Walter thought about this. If we make brains bigger, if we just add more neurons, there's a problem because the connectivity increases non-linearly on top of that. And so then it almost implies by necessity, we have to disconnect the brain more. As we make bigger circuits, we have to have fewer connections. This is again something that deep nets do not face because they can be infinite, they can be n square. We don't care, right? But the brain cares about that because if your brain doesn't fit, that's not good. So there are these scaling relationships to do with body size, but then these relationships to body plan and behavior that we're just about to dig into, as we have now available to us something like 70 or 80 different mammalian species.
Fascinating, Olaf. Thank you. Clearly, we're just at the tip of the iceberg. It's very extremely exciting. I